Thank you all for joining us today for this introductory webinar to the AmpliSeq for Illumina workflow. Today, we will be going over the AmpliSeq workflow and available products for the kit options, options for content selection and customizing panels with Design Studio, and some of the available analysis options. Before diving into the workflow details, I want to give you a very quick overview of the process. AmpliSeq is an efficient workflow with very little hands-on time. The library prep is just 1.5 hours of hands-on time. So this is 10 steps that take about 5 to 7 hours in total. The next day, on day 2, you can move forward with sequencing on one of our easy-to-use sequencing platforms. And then on day 3, you're ready for the analysis, either locally or in the cloud. So that's great. AmpliSeq is quick and easy to use. But how do you know if it's a good fit for your project? AmpliSeq is a type of targeted sequencing where by generating libraries for only the regions of interest, we're able to focus and concentrate the sequencing output to enable deeper coverage of more specific areas. This workflow consists of Multiplex PCR to selectively amplify genomic regions targeted by the oligo panel. We then add adapters and amplify the libraries. AmpliSeq panels can be used to target a wide range of regions, from 12 amplicons up to thousands. Either DNA or RNA is compatible, including high quality or degraded input, even FFP samples. We have ready-to-use or pre-designed panels that may suit your needs, and if not, we offer custom design panels as well. These include both DNA and RNA custom panels, including for non-human species. We will talk more about all of these topics shortly. Here's a visual overview of the key steps in the AmpliSeq library preparation. DNA, or cDNA, generated from reverse transcription of RNA input, is amplified with forward and reverse primers in the panel. This is a multiplex PCR reaction with many primer pairs in the same pool. Some panels will require multiple pools to avoid collisions, creation of large amplicons, and avoid inhibitory primer interactions. The design process and strategies employed by the design algorithms enable the success of this highly multiplexed approach. Once we have amplicons, the primer regions are removed, and Illumina sequencing adapters are added via blunt end ligation. The final libraries are amplified, and we're ready for sequencing. Now that you have an overview of the process, let's go into more detail on each step and learn about the various kit options that are available. Required library prep reagents include a Library Plus kit, a panel of your choosing, and an index adapter kit. There are also some optional accessory kits available that we will cover later. The Plus kit consists of enzymes, buffers, and PCR mixes that are needed for library prep. The panel contains the oligos needed to target amplification to the regions of interest. And the index kit enables multiplex sequencing and compatibility with Illumina sequencing platforms. All AmpliSeq index options are plated and pre-mixed. We can multiplex up to 384 samples using the CD, that's combinatorial dual indexes. The large volume CD index kit has a higher fill volume for convenience when using exome panels or automation with panels of 4 or more pools. Currently, there's only a 24 index UD unique dual index kit available at this time. Those are 8 base pair long indexes. Please note that the shipping and storage temperatures are different for the panel and the indexes. So you might receive a shipment at room temperature, but it needs to be stored at -20. So please always refer to the storage temperatures listed on the packaging. A quick note about the different panel options. We're going to go through this slide and these categories in more detail later when we talk about content selection. For now, I just want to point out that it's important to decide what panel will be used very early in your planning process, since different panels are split up into a different number of amplicon pools. And the number of amplicon pools per panel will affect what kits are needed. So you'll purchase the Plus reagents separately from the panel. The number of reactions you can get out of a given plus kit depends on how many pools are in your panel. This chart displays the number of libraries that can be prepared with each size library plus kit. These come in 24 reaction, 96 reaction, and 384 reaction sizes. Again, note how the number of libraries that can be prepared with each size plus kit depends on how many pools are in the panel. For panels that accept DNA and RNA input, DNA and RNA are prepared separately. So from one biological sample in which DNA and RNA are isolated, you would actually be generating two libraries, one from the DNA and one from the RNA. So please take this into account when you're calculating how many libraries you will be preparing using this table. The specifications for how many pools are used in each panel can be found on the panel product page, or when you have completed a custom design. Additional information can also be found in the reference guide. In addition to the Library Plus kit, the panel, and the indexes, there are also several optional kits that may be needed in some situations. Again, it's good to be aware of these requirements when you're planning your project. We will go through each of these accessory products in more detail shortly. So now that we have a sense of what reagents are available, let's talk through the AmpliSeq library prep workflow step by step. Here on the left, you can see we have the schematic diagram with a lot of Eppendorf tubes showing the various key steps we'll be going through. So to get started, let's discuss how to prepare input. We will first need to quantify and dilute the DNA. If you're working with RNA, that needs to be converted to cDNA with the AmpliSeq cDNA synthesis for Illumina kit. Again, please pay attention to how many pools are in your panel, and note that the input guidance is provided per pool. AmpliSeq chemistry generally works well with FFP and degraded input, but check the panel reference guide for specific recommendations. Panel Amplicon sizes can be optimized for degraded input. Generally, we want shorter Amplicons if the input is going to be highly fragmented. The library yield can end up being a bit lower for degraded library samples, such as FFP DNA. Also, highly degraded input may not result in full coverage of the target regions, as the primers need to be able to sit down on both sides of the target region. With that said, there's no formal limit on the quality of input for AmpliSeq. Once the input is prepared as specified in the reference guide, we simply set up PCR reactions with a separate tube for each primer pool. The hi-fi mix comes with the kit. We recommend creating a DNA or cDNA master mix for each sample, and then aliquoting that DNA master mix into separate tubes for each primer pool. You then add the panel pool primers as instructed. For the PCR program itself, we can tailor the cycles and the extension times as needed. Generally, the more primer pairs are in the pool, the fewer cycles we will use, but the longer the extension time will be. Please refer to the specifics in the reference guide. You can decrease the number of cycles if using higher input, or increase the cycles if using lower input amounts. For degraded input, increase the number of PCR cycles by 3. There is a table in the user guide shown here for how to modify the number of cycles or the extension time, depending on which of these conditions apply. Note that these changes are additive, so if multiple situations apply for your specific project, you can keep a running tally of the cumulative recommended adjustments. For most troubleshooting scenarios, we can start by increasing up to 5 cycles for the target PCR step. So once we've amplified our targets in multiple pools, we can then combine them together into one tube for the rest of the protocol. We then chew back the primers enzymatically with FUPA, leaving our insert of interest. In the diagram below, you can see we start with the primer regions on the Amplicon, which are removed after digestion, leaving the insert and about 1 or 2 bases of the primers left on the end. This will create blunt-ended phosphorylated fragments. Next, we will set up ligation reactions in the same well. Which solution sets up the ligation conditions, and ligase is used to attach the indexes. Note that only AmpliSeq adapters can be used with this protocol. No other Illumina indexes can be used. Be sure to pre-plan your index pooling strategy based on how you will be performing the sequencing run to ensure for proper index combinations if you are sequencing only a few samples at a time. The index adapter pooling guide has guidance for which index combinations to choose when working with low plexity pools in order to ensure color balance. Generally, if you pool down a column of the CD index plate, this will give you unique dual index combinations if you're working with a low plex pool, but you can also check out the index adapter pooling guide. So now that the adapters have been added, we need to do a quick cleanup before the final PCR. This is done with user-supplied Agincore Ampure XP beads at a 1:1 ratio. The 1:1 ratio should remove any small fragments less than 200 base pairs in size. Because ethanol can inhibit PCR reactions, after the ethanol washes, it is better to slightly over-dry the beads than risk carrying over ethanol into the next step. The final library is amplified with a quick 7-cycle PCR. Note that the beads from the previous 1x bead cleanup do remain in the well for the PCR reaction. After we've amplified the library with that PCR reaction, the libraries will go through another bead cleanup. This time, though, it's a double-sided cleanup. So we'll do a 0.8x ratio cleanup to remove the large DNA, and then a 1.2 ratio to remove the small fragments. During this process, it's critical to remember which fraction is being discarded or kept for each step. For new users, it may seem a little bit odd, but you will be adding even more beads in addition to what's already in the well. Specifically, you add 25 microliters of Ampure XP beads to each well containing about 50 microliters of library. Then seal the plate and vortex. This step is adding beads to the beads that are already in the reaction. After the magnetic stand has pulled the beads out of solution, the supernatant is kept and moved to a new well. Then we add more beads, seal the plate again, vortex, and place on the magnetic stand again. This time, though, we will remove the supernatant and keep the pellet. 2 washes, then we elute the final cleaned libraries in low TE buffer. To finish out the library preparation process, we need to check the library quantity and quality. We recommend the fragment analyzer or adjuvant bioanalyzer. Most libraries will have a size distribution from about 200 to 275 base pairs. Of this, about 135 base pairs is the adapter content. Note that each panel will have a different size distribution. For Amplicon libraries, we typically think of a single sharp peak from a single Amplicon, but remember that these are large multiplexed pools, so we actually will end up with a broader peak. To quantify AmpliSeq libraries, several different methods can be used. You can do fragment analysis with a fragment analyzer or bioanalyzer, a fluorometric method such as qubit or a quantit picogreen assay, and also qPCR, such as with the Kappa library quantification kit. So these are all acceptable methods for AmpliSeq libraries. To get the final libraries ready for sequencing, you'll need to convert the concentration measured from nanograms to microliter to nanomolar using the size of the final library to do this conversion. From there, we'll want to normalize all samples to 2 nanomolar. You can use the pooling calculator linked here to assist with the math, always being sure to avoid pipetting small volumes, as these measurements can be less accurate with your pipetter. For additional guidance, refer to the panel reference guide and the Denature and Dilute Libraries guide for the Illumina sequencing platform that you will be using. We do have some recommendations for how much to load on different sequencers for specific panels. These are intended to be a reference point to get you started. So you may find you need to further optimize the loading concentration to really dial it in with your quantification method in your specific sequencing instrument. The second table shown here is from the Denature and Dilute Libraries Guide and shows the recommended pooling ratio between DNA and RNA libraries for panels where both DNA and RNA is used. So if you're using one of those panels, please again refer to the Denature and Dilute Library Guide for that information. Since AmpliSeq libraries are highly targeted, any of our benchtop systems are a great choice for sequencing, including the iSeq, MiniSeq, MiSeq, NextSeq 550, and NextSeq 2000. When deciding on a read length, keep in mind that overlap is generally desirable for most workflows. For most of our fixed panels, 2x150 cycle paired end sequencing is recommended. For the number of samples per run, please see the product compatibility information, which can be accessed from the Library Prep and Array Kit Selector tool or from the appropriate support page. For custom panels, you can choose a read length appropriate for the length of the Amplicons in your design. For more sequencing details, please refer to the 2 Amplicon sequencing webinars that are linked here. Now that we've gone through the typical AmpliSeq workflow from input to sequencing, we will briefly go through a few optional workflow enhancements to conclude this section of the talk. The library preparation process can be automated using various automation equipment. Currently, Beckman Coulter, Eppendorf, and Perkin-Elmer have listed protocols, and other vendors also have protocols in the pipeline. Please see the library preparation automation page for more information. As promised earlier, I now want to return back to these optional accessory products I quickly showed you. First, the AmpliSeq cDNA synthesis for Illumina kit. This kit is required for making RNA libraries, as we first need to convert the RNA to cDNA using these reagents. Next, the AmpliSeq for Illumina sample ID panel. This is optional for DNA libraries and includes several human SNP primer pairs that can help validate sample identity. In the purple box, we have the AmpliSeq for Illumina Direct FFPE DNA kit. This is an optional kit for FFPE DNA samples. The kit provides sufficient reagents to bypass nucleic acid isolation and prepare DNA from unstained, slide-mounted, formalin-fixed paraffin-embedded, or FFPE, tissue samples. A standalone reference guide for this process is available on our webpage, and this would be done before beginning the AmpliSeq workflow. Lastly, the AmpliSeq library equalizer for Illumina. This is a bead-based normalization that can be used either for DNA or RNA libraries. Instructions for use are in the appendix of your panel reference guide. Be sure to follow the appropriate protocol in your instrument's Denature and Dilute library guides when working with libraries normalized using the equalizer kit. A few more usage notes on these kits. Again, I think the cDNA synthesis kit that's used for converting RNA to cDNA is pretty self-explanatory. It comes with enough materials for 100 to 200 RNA reactions, depending on the panel, because some panels will use more input volume than others. AmpliSeq for Illumina Direct FFPE DNA kit. This is only for use with AmpliSeq for Illumina protocols. The library equalizer kit. So note that while this kit is compatible, manual normalization rather than the equalizer workflow might be preferred for some panels with less consistency in yield. For example, exome or immune repertoire panels, in which we might expect to have more challenging sample types. So it's compatible, but use it at your discretion. Note that if you want to use the equalizer protocol, you need to decide this before starting the workflow. The pink box here is outlining kind of a fork in the workflow protocol. So you need to decide this before you get to the end of the library prep. The reason for this is that you have to add additional PCR cycles at the Amplify library step if you're going to be using the equalizer workflow. So again, this isn't something you can just decide to do at the very end, as it'll already be too late to make the needed protocol adjustments. If you do use the equalizer beats, the resulting libraries will be double-stranded DNA. The libraries are then pooled by equal volume and prepared for denaturation. Again, please refer to the appropriate section of the denature and dilute guides for our benchtop instruments for suggested loading volumes. Lastly, the sample ID panel. Again, this is intended to provide added assurance that there's been no mixing or mixing up of the samples. The specific SNPs targeted are detailed in the sample ID application note, specifically table 2, which is shown here. Since these are human SNPs, this optional kit would only really be useful with human samples and panels. The sample ID panel content is already included within select AmpliSeq for Illumina panel, including the BRCA panel, the comprehensive V3 panel, and the myeloid panel. If used, the sample ID panel is spiked in at the Amplify DNA target step of the workflow. So now that we know how to execute the assay, let's discuss how to select the desired content for your project. If you want to target custom content, you can use our Design Studio software. Before embarking on designing a custom panel, you may want to first check and see if there is a ready-to-use or pre-designed community panel that might already have what you need. As a reminder, here are the 3 types of AmpliSeq for Illumina panels I briefly displayed earlier. The AmpliSeq for Illumina ready-to-use panels are pre-designed, synthesized panels that are available in small reaction sizes, usually 24 reactions. AmpliSeq for Illumina community panels have been pre-designed with input from leading disease researchers and are made to order, so these come in larger reaction sizes. And of course, the custom options, which we will get into shortly. First, let's go over the specific ready-to-use panels that are available. Each of these panels can be found on the Illumina website, where there's demo data on base space, data sheets, and product files such as manifests and bed files that detail the panel contents. If you have specific genes of interest and want to see if these panels include those targets, you can try the Gene Panel and Array Finder tool on our website. We can break down the ready-to-use panels into 3 categories based on compatible input type. There are DNA panels, RNA panels, and panels where DNA and RNA can be used. Again, more information and the specific contents of each of these panels can be found on our website. The next category of panel options are the community panels. Illumina community panels have been pre-designed with thought input from leading disease researchers. Note the number of pools when ordering the panel, as this affects, again, how many AmpliSeq Library Plus kits are needed. The number of tubes supplied with community panels will change depending on if it's DNA or RNA, the number of Amplicons, and the number of primer pools. But each tube will contain 1,500 microliters. We currently have 32 community panels available, and you can browse these on our website. Targets for community panels include human, viral, and bacterial options. For example, we have the AmpliSeq SARS-CoV-2 panel, a panel that includes regions of interest for antimicrobial resistance, inherited cancer research panels, and many more. Each panel has a specific product page where you can find important information about the panel, including the specifications table that details the number of Amplicon pools and input amounts that are needed, and the product literature section, which is where you'll find the manifest with the target list. For all community panels, you'll need to use the AmpliSeq for Illumina on-demand custom and community panels reference guide. For custom panels, the options differ somewhat between DNA and RNA panels. Both DNA and RNA panels can be designed with Design Studio. For DNA panels, there are a variety of genomes to choose from and various options for full custom, on-demand, and on-demand spike-in options, which we'll discuss shortly. For RNA panels, human transcriptome options are available, and there are expression panels and gene fusion panels. We will talk about each of these types of custom panels, starting with the DNA custom panels. Each of these 3 custom DNA panel types can be created in Design Studio. Hotspot panels consist of a single pool with no overlapping amplicons and are intended primarily for SNPs and small indels. Consider if your target could be covered by a single amplicon, or if a single primer pool could cover the entire region of interest. Gene panels are intended for genes and larger target regions. These default to a 2-pool design, but may have more than 2 pools. Gene panels use multiple primer pools to make sure that neighboring primer pairs do not interfere with one another. So in this example here, the orange and purple primers could cause undesirable off-target amplicon products if they were in the same primer pool together. So they've been separated into 2 primer pools for the PCR reaction. For both hotspot and gene designs, you can have 12 to 3072 amplicons per pool. Note that if you have content larger than about 5 kb, you might instead consider an enrichment design instead of using amplicons. Lastly, the on-demand panels. These are distinct from the hotspot and gene panels because the available content has been pre-tested in a wet lab environment. These are specifically coding regions, not UTR or introns, and there are over 5,000 pre-tested genes to consider. In addition to getting the amplicons, you also get performance data for these genes. On-demand options can be combined with the other custom DNA panels if only some of the genes you are interested in are available on demand. This is what we refer to as an on-demand spike-in panel, since you are spiking in additional custom content. We will discuss specifically how to design these custom DNA panels in Design Studio shortly. Now onto the custom RNA panel options in Design Studio. We have RNA expression and RNA fusion panels. RNA expression panels are pre-designed amplicons that have been evaluated through the AmpliSeq transcriptome human gene expression panel. This is a customizable expression panel, so you can add or remove genes as needed. RNA fusion panels can be designed by selecting from a predefined list of assays for fusion and gene expression. You can also create mixed fusion expression panels. Now that we have some context for the various custom content options, let's go through how to design a custom panel in Design Studio, starting with an example, DNA custom gene design. Much more information about Design Studio is available in the recorded webinar that's dedicated specifically to this topic, Design Studio Introduction. And you can find that on our YouTube channel of recorded support webinars. So for this demonstration, we'll select DNA for the assay type at the top, and then we'll do AmpliSeq for Illumina gene, as opposed to the hotspot, which is, again, remember, single pool, or the on-demand, which are those wet lab tested. We'll pick the species and the reference genome for our samples. In this case, we're going to do human HG19, the selection on the far left. Note that for DNA panels, extended species are also available to choose from. At the bottom of the screen, enter a name for this design in the system. Once we've chosen our species and design name, we can then choose the sample type in the amplicon length. The sample type selected will limit the available maximum amplicon length options. So it's kind of like a filter. The regular sample type is for high quality input. The other options are FFPE or cfDNA for highly degraded input. The more degraded the DNA is, the smaller the maximum amplicon length should be to give the best success with the panel. For cell-free DNA or cfDNA, the only available option is 140 base pairs, since the input is very degraded. Whereas if you're working with regular high-quality input, all 4 maximum lengths are available. Now you're ready to input targets, and for that we have a few different options for how you can enter those. By gene name, by genomic coordinate, or you can enter a large number of targets with a file template. To enter genes, enter the gene name and decide if you want the coding sequence only or the exon only. Exon only includes the coding sequence along with the 5' and 3' UTRs. Throughout Design Studio, there are several question mark icons that you can click for definitions of terms. If you want to include introns rather than the Add Gene option, you can input targets by coordinate. For the chromosome field, be sure to use the letters CHR and then the chromosome number. Also, take care to input the coordinates in the reference genome build that corresponds to the selection you initially made at the beginning of the design workflow. So you recall where you're selecting a species, there were different options for human HG19 versus GRCH38 and other species as well. So make sure that that selection that you made initially, you're going to be entering the coordinates in that genome build. You can also upload a file. For larger designs, you may want to download the template, enter all of your input, and then upload that file to add all the targets at once. You can see here there's the option to download the template and then upload it back into the system. The Copy by Amplicon feature allows you to copy existing Amplicons either from a previous design or from a community design. Again, you can download a template, modify, and then upload the file. Be sure that when you're uploading this file that you've included the Amplicon ID and the solution ID that's required in the template. Lastly, you can add Amplicons from a community or fixed panel. Simply select the Browse button, and then you'll see a list of those community and fixed panels that you could pull Amplicons from into your own design. Here are some examples of what those last two options look like. So on the top, we have the Copy by Amplicon template. Again, be sure to include the Amplicon ID and the solution ID. The second section of this slide shows how it will look if you choose to add Amplicons from community and fixed panels. You can browse the available panels and copy Amplicons as desired. Great, so once we've made all of the selections and have submitted the design in Design Studio, you'll get an email when the design is completed. You can then follow the link provided and you'll have the opportunity to review the design, which is what you're seeing here. You'll get a report summary at the top that shows you the calculated in silico coverage by the designed Amplicons and the parameters of your design, such as the total target size, the number of pools, how many Amplicons there are per pool, the minimum, maximum, and average Amplicon length. You can see there's a lot of metrics covered in this design summary. The See More Solutions link allows you to review alternative designs. The algorithm by default will automatically select the best design for most users, but if desired, you can adjust the stringency. You can also modify your design, which essentially copies the current design and allows you to add or modify targets. The current design will remain as is in your account, and the modifications will occur in the copied design. You can go into each target or gene and see the specific regions of coverage if you just click on that little pop-out to the UCSC genome browser in each row of the table. And this way you can see specifically where you have Amplicons and where you have gaps to better determine if this impacts your experimental goals. Lastly, I just want to draw your attention to the Copy, Transfer, and Delete buttons in the upper right corner. This concludes the AmpliSeq for Illumina gene demonstration. The hotspot option in the center of the assay technology selection is very similar, so we're not going to go through that right now. However, let's quickly go through the on-demand option. Recall that these are the wet lab tested gene designs that have performance data available. The only species and genome available with this option is human HG19. To manage targets for the on-demand options, you can search the available on-demand content by gene symbol. If your gene of interest is not available on demand, use the Spike In option to add it. There are over 5,000 predefined genes to choose from on demand. Note that while individual genes have been tested, not necessarily all combinations of all genes have been tested. These target just the coding sequence. Introns and UTRs are not included. Typically, these are for smaller reaction sizes, so 24 or 96 samples. If a desired gene is not available on demand, you will see this warning message informing you that Genome's Design Studio has converted it to a Spike-In. On-demand and Spike-In content can be combined. So that concludes the demonstration of Design Studio for this webinar. I want to finish out this section of the talk by providing you with resources to help with several frequently asked questions. One question we commonly get is, how many samples can I sequence? To make an informed decision about this, we suggest using the sequencing coverage calculator on the Illumina support site. There is also a short video explaining quickly how to use this tool on our YouTube support short channel. From the calculator, you will select whether you are using a DNA or an RNA panel, pick it from the list, and then input additional information about the read length and desired coverage to ascertain what the maximum number of samples are that can be combined in a single run, given the sequencing platform and sequencing kit that will be used. So this is a really helpful tool to help answer this question. Once you have a finalized custom or on-demand panel ready in your Design Studio account, you can help figure out which products are needed using the estimation tools that are shown here. This just enables you to further plan your experiment. From the Illumina website, you can log on to My Illumina and then navigate to Tools, Custom Products, AmpliSeq for Illumina. From here, you can select the panel type you are working with and check the box to include recommended consumables, then populate the intended sequencing platform and reagent kits. From here, you can print the estimates that are provided, request a quote, or add to the cart directly. So this can be a helpful tool since there are so many different accessory kits available and different sizes depending on how many pools and reactions you have. Another question we get asked a lot is where to find the relevant product files for your AmpliSeq panel. For ready-to-use panels, the manifest is available on the product page under the Manual and Support Information section, following the path listed on the slide here. For community panels, the manifest is also on the product page under the Product Literature section. For custom gene and hotspot designs, the manifest is available through Design Studio, but please note that the design must be in the designed state for the manifest to be available. Custom on-demand panel manifests are available after ordering. The manifest is an important document for analyzing your data, so it's important that you know where to find this and have it ready. So what is a manifest? This is a text file that lists out all of the targets in the AmpliSeq panel we are using. So even though they're text or TXT files, they can be viewed in Microsoft Excel instead of a text editor, since this will help allow you to see the proper formatting, like how the columns are supposed to align. So let's discuss now how the manifest is used during analysis. Since this is an introductory webinar, we will be doing a high-level overview of the available analysis options. There are both online and offline analysis options available. Amplicon analysis tools work by aligning the reads from the sequencer to a reference that is specified in the manifest. Analysis outputs vary depending on the type of panel, but generally they can call variants, assess coverage, identify differentially expressed genes, and call fusions. Each panel will have several different expected metrics, depending on the panel design and sequencing. In this diagram, you can see we have several base space sequence hub applications that can be used to analyze Amplicon data online, and we also have offline options, either on a Dragon server or in local run manager. Depending on the analysis workflow, the input for analysis might be fast queues, or it could be the full sequencing run folder. Please refer to the specific documentation for your analysis workflow of choice for details on the required input for analysis. Regardless of the specific analysis option you choose, it's important to understand key analysis metrics. Since the DNA Amplicon analysis is the most commonly used, we're going to go through each of these key metrics that you may encounter in your analysis results. Alignment, which is how well the data is aligning to the reference genome. On target, which is the % of the data aligning to the target regions in the manifest, as opposed to off-target regions. Coverage, which is the sequencing depth for each amplicon. Dropouts, so this shows if there are any amplicons with 0 reads. And uniformity, which is how even the coverage is over all of the amplicon targets. So those are great definitions, but let's quickly visualize what each of these metrics mean. First, we need a reference genome. The genome will be used as our reference sequence, to which we align our sequencing data, or our reads. Here, I have added reads to our schematic. The aligned reads align to the reference sequence. Unaligned reads do not align to the reference sequence. On-target reads will align to the regions defined in the manifest file. So these are the regions that we wanted to sequence with the panel. Off-target reads will still align to the reference genome sequence, although they do not align to the regions that are listed in the manifest file. So these are the reads that are not within the target area of the Amplicon panel. You can see here why the manifest is an important document for the analysis. Coverage refers to the number of reads for each panel target, or the reads per amplicon. Amplicon targets in the manifest that do not get any reads are known as dropouts. For our coverage uniformity calculation, we look at the reads for all amplicon targets and compare them. This metric will be calculated for you during the analysis. For us to review the analysis results, we need the analysis output files. If the analysis was performed with local run manager, the output files will be located in the alignment folder in the analysis output location. If the analysis was performed in BaseSpace, the analysis files will be in the Files section of the BaseSpace project. For more information on what to expect within the output files, please see the recorded webinar, Amplicon Sequencing, Assessing the Success of Your Run. If you have any questions regarding where to find analysis files or how to interpret them, please reach out to us in technical support. To wrap up now, we will quickly go over some additional resources. I've included links here for more information on the AmpliSeq library preparation process, the panels, and custom panel design. There are also links to several more recorded webinars that you may find helpful. And here are just some additional resources with links for FAQs, support pages, and additional online training resources. I especially want to call out the last 2 items here, which are application notes, one for the sample ID panel, and one that can be useful if you're working with FFPE samples. So thank you so much for taking the time to join me today. I hope this session has been helpful for you.